To understand the mechanics of blood clotting, we first have to look at the circulatory system as a highly pressurized, closed fluid network. This system continuously drives roughly 1 trillion individual cells through the body under immense arterial force. When a physical tear occurs, the body faces an immediate mechanical paradox. It must trigger a localized solidification to seal the leak within 15 to 120 seconds, without allowing that exact same process to trigger a runaway hardening of the entire fluid supply. The first line of defense is a rapid mechanical reaction known as a vascular spasm. The physical trauma to the vessel wall, combined with sudden pain receptor reflexes, forces the surrounding smooth muscle to rapidly contract and choke off the blood flow. This localized constriction buys the system a few critical seconds, but it cannot permanently hold back continuous arterial pressure on its own. The actual physical repair begins far away from the injury site, deep inside the bone marrow. Here, massive precursor cells called megakaryocytes continuously fragment their own cytoplasm to produce platelets. These are not full cells, but tiny enucleated discs, measuring just 2 to 4 micrometers across. These platelets circulate passively until they make direct physical contact with subendothelial collagen fibers, which are normally hidden, but become exposed the moment a vessel wall tears. That single point of contact forces the platelets to swell into irregular shapes and secrete specific chemical signals, namely ADP and thromboxane A2. These secretions alter the surface properties of nearby platelets, making them highly adhesive, so they instantly stick to both the exposed collagen and to each other, forming a temporary physical plug. This initial cluster stops the immediate bleeding, but the structure is physically fragile. To survive the long-term shear force of flowing blood, the body initiates a precise biochemical cascade to build a reinforced matrix over the gap. The permanent seal is constructed through the coagulation cascade, a self-amplifying loop of enzymatic reactions. This flowchart maps the extrinsic pathway, an explosive reaction chain initiated strictly by trauma outside the blood plasma. Crushed perivascular cells release a lipoprotein called tissue factor. When exposed to circulating calcium ions, this tissue factor instantly activates factor VII. Because it relies entirely on massive external cell damage, this extrinsic sequence operates at extreme speed, capable of initiating a clot in as little as 15 seconds. By contrast, this diagram traces the intrinsic pathway, a slower amplification sequence triggered strictly inside the bloodstream. This internal trigger sets off a domino effect. Factor 12 activates factor 11, which activates factor 9. Finally, this complex process activates factor X. Despite relying on entirely different environmental triggers and operating at different speeds, both of these biological algorithms mathematically converge on a single critical molecular node, factor X. This diagram shows the final shared sequence known as the common pathway, which strictly begins the moment factor X is activated. Activated factor X combines with factor V, platelet phospholipids, and calcium to form an entirely new enzyme complex called the prothrombin activator. Throughout this entire sequence, calcium ions operate as the essential binding agent that allows these protein complexes to assemble and function. The prothrombin activator immediately cleaves an inactive plasma globulin called prothrombin, transforming it into a highly active proteolytic enzyme known as thrombin. Thrombin's specific function is to target massive, high molecular weight fibrinogen compounds circulating in the plasma, shearing off peptides to create smaller, active fibrin monomers. These new fibrin monomers automatically polymerize into long threads, while factor 13 cross-links them into a dense, interwoven structural reticulum. Through this exact sequence of enzymatic cleavages, the fragile temporary platelet cluster is successfully converted into an insoluble, pressure-resistant physical barrier. Once this explosive amplification loop begins, it generates enough thrombin to potentially clot the entire circulatory system, so the body requires active, immediate brakes. Now, the very first failsafe is purely physical. The smooth endothelial glycocalyx lining the inner walls of healthy blood vessels naturally repels circulating clotting factors and platelets. As the clot forms, excess thrombin binds to an endothelial protein called thrombomodulin. This activates a specialized molecule known as protein C. Protein C acts as a biological assassin, inactivating factors V and 8, physically choking off the amplification loop. Meanwhile, an alpha-globulin called antithrombin 3 continuously scrubs the blood, actively adsorbing and neutralizing any free-floating thrombin over a 12 to 20-minute cycle. This scrubbing process is accelerated by heparin, a compound continuously released by mast cells in the surrounding connective tissue. Human survival depends entirely on these active, calibrated inhibitory networks running in exact parallel equilibrium with the coagulation triggers. If even a single variable in this algorithm goes missing, the precision of the entire hemostatic system fails. For instance, a genetic deficiency in producing factor VIII results in the condition known clinically as classical hemophilia A. Without factor VIII, the intrinsic pathway breaks down. The blood can no longer effectively activate factor X, which completely stops the common pathway from initiating. A more systemic disruption occurs in the liver. Severe liver disease, or a strict deficiency in vitamin K, limits the organ's ability to synthesize baseline levels of prothrombin. With diminished prothrombin available to convert into thrombin, coagulation times stretch from mere seconds out to 10 minutes or more, exposing a patient to massive hemorrhage from minor internal injuries. Hemostasis is a highly conserved zero-tolerance biochemical algorithm. The absence of a single targeted protein transforms a daily, life-saving physiological response into a catastrophic systemic failure.